Hi, my name is Shinny Pimlapas. I'm from the Technical University of Denmark, or DTU. I'm going to show you one of our work on the effect of population's diet to antibiotic resistance via metagenomics analysis. So this work actually came from the question that can we actually measure the change of AMR because of the diet, and can we measuring that in the population level when the whole population or the majority people change the diet? Is the effect to the AMR or effect to the bacteria? So that's the main question of our work on this project, and we actually took a sample from a place where the majority of people change their diet willingly. So that place is Thailand. So we took a sample from 3 cities in Thailand: the north, the Bangkok, and the South Surat Thani. And those cities, the people there, they're very much participating in what we call vegetarian festival, that 10 consecutive days that people change the diet from omnivores to be vegetarian. And that happened in October 2019, where we took the sample. And the sample that we took is a sewage sample that could represent what's going on in the population. So we took a sample a month before the festival, half a month, and a few days before the festival. And we took a sample every single day through 10 days of the festival. We also took a sample a few days after, half a month, and a month after the festival. And what we did with the sewage sample that got taken, we did extract all the DNA and do metagenomic sequencing, and we did metagenomic analysis. And first of all, we mapped the metagenomic sequences to bacterial database. And what you can see here, that the bacteriome or microbiome of this sewage sample from 3 cities, as you can see here, you can see that the red circle of all of this is the sewage sample from the sample during the festival. So the microbiome of the sewage sample during the festival is different from the before and after the festival. So we can see already the change or difference in terms of bacterial profile of the sample during the festival. And looking at the AMR, looking at the total abundance of AMR in the sewage sample, this is the city from the north, Bangkok, and from the south. And you can see that the line between here, I draw it between the sample before the festival, during the festival, and after the festival. And you can see that the total abundance of AMR actually increasing for the northern city and Bangkok when the festival started. We see different thing from the south, that there is a decrease of abundance of AMR after the festival ended. And that changes, we also did some statistical analysis called change point analysis to see if that change is statistical significant, and it actually does. You can see this is the change point in the north, Bangkok, and the south, and the change point is actually happen when the festival starts in both the north and the Bangkok, and also changes in the end of the festival for the south. So not only the microbiome that it changed during the festival, and also the abundance of total antimicrobial resistance. And not only that we tested, we actually mapped the metagenomic sequencing data into what we call KSAM database. The KSAM database is a database containing genes related to enzyme. And what you can see, this is abundance of the gene related to enzyme in different functional categories from the north city, Bangkok, and from the south. What I want to point out here, the second most abundant enzyme in the dark blue here, that increasing when the festival starts for Bangkok and from the north, and then it's decreasing after the festival ended. This dark blue here is the gene that related to carbohydrate metabolism. So not only that the abundance of AMR increasing when the festival starts, and also with the carbohydrate related genes also increasing when the festival starts, and that festival is related to vegetarian festival. We don't know, we don't have an answer for you why these changes happen, but what we can conclude from this work right now so far is that yes, there is a change of the antimicrobial resistance and microbiome when people change their diet. And we can capture that change in the population level through the sewage data, through metagenomics analysis. And that's all, like a very sum up of our works, and thank you for watching.